everyone, my name is Vienna Protomastro. Today's date is April 19th, 2024, and today I will be doing a presentation on the anatomy and enzymology of fat absorption. Alright, so fat absorption is a crucial physiological process for obtaining energy and essential nutrients from dietary fats. It involves the breakdown of fats into smaller molecules, the transportation across the intestinal wall, and the subsequent metabolism and storage of fats within the body. And so digestion of fats begins with the secretion of bile by liver cells, which are known as hepatocytes. Bile is a greenish-yellow non-enzymatic fluid that is secreted, again, by hepatocytes. The key components of bile are bile salts, bile pigments, and cholesterol. Bile salts facilitate enzymatic fat digestion, and bile pigments such as bilirubin are the waste products of hemoglobin degradation, and cholesterol is excreted in the feces. So bile salts, which act as detergents to make fats soluble during digestion, are made up of steroid bile acids combined with amino acids. And this image on the right of the slide, we can see bile through an endoscope, which is pretty cool, and you can definitely see the greenish yellow coloring of bile pretty clearly here. Alright, and so this image is from our textbook Silverthorne. So bile produced by hepatocytes in the liver is transported through the bile ducts and stored in the gallbladder. And upon ingestion of a fatty meal, contraction of the gallbladder sends the bile into the duodenum through the common bile duct. The duodenum is the start of the small intestine, and this is where bile salts will begin to break down fats. Alright, and so fats are not water soluble, which can make it pretty difficult for enzymes to access and break down dietary fats effectively. So bile salts act as emulsifiers, which means they have both hydrophobic and hydrophilic regions. And when bile salts come into contact with fat globules in the digestive tract, they surround the fat molecules with their hydrophobic portions interacting with the fat molecules and their hydrophilic portions interacting with water. And this process breaks down large fat globules into small fat droplets, and this process is known as emulsification. Alright, and so by increasing the surface area of fat exposed to the two digestive enzymes, emulsification facilitates the action of enzymes like pancreatic lipase, which can then more effectively break down fats into smaller molecules that can then be absorbed by the body. And they do this by removing two fatty acids from each triglyceride molecule. This results in one monoglyceride and two free fatty acids. The bile salt coating, however, can complicate digestion because lipase is unable to penetrate the bile salts. Therefore, digestion of fats also requires something called colipase, which is a protein cofactor secreted by the pancreas. And colipase displaces some bile salts, which allows lipase to then access the fats inside the bile salt coating. Alright, and once the fats are broken down into these smaller molecules that we discussed previously, they then come together to form small disc-shaped micelles, which enter the aqueous layer at the edge of the brush border. And again, these small disc micelles contain bile salts, phospholipids, fatty acids, cholesterol, and mono and diglycerides. Alright, and so this diagram represents the simple diffusion of monoglycerides and fatty acids moving out of the micelles and entering the cells via simple diffusion. And so yes, monoglycerides and fatty acids move across the enterocyte membrane using simple diffusion, meaning they do not require any specialized channels or carriers or anything like that. Cholesterol, on the other hand, is transported across the brush border membrane using energy-dependent membrane transporters, specifically one called NPC1L1, which is the protein that is inhibited by ezetimibe, and ezetimibe is a drug that inhibits cholesterol absorption. Alright, and so once monoglycerides and fatty acids enter the enterocytes, they move into the smooth endoplasmic reticulum, where they undergo recombination into triglycerides. The triglycerides then join cholesterol and other proteins to form large droplets called chylomicrons, which we can see here in the diagram on the right of the slide. And due to their large size, chylomicrons are packaged into secretory vesicles by the Golgi, and then they leave the cell via exocytosis. All right, and so on the right is a blown up photo of the diagram we saw on one of the previous slides. So since chylomicrons are very large, this also prevents them from crossing the basement membrane of capillaries. So instead, chylomicrons are absorbed into lacteals, which we can see here on the right, that lacteal, which are specialized lymphatic vessels in the small intestine. Chylomicrons pass through the lymphatic system and enter the venous blood right before it flows into the right side of the heart. Some shorter fatty acids are not assembled into chylomicrons and can therefore cross the capillary basement membrane and go directly into the blood. All right, and that is the end of the process of the anatomy and enzymology of fat absorption. So I created a study guide to help, study for our next exam and go over some of those terms that we discussed in this presentation. So if you want to take a second and pause the presentation to try to match the term to its correct definition, feel free to do so. Right, and here are the answers to the study aid. I found this very helpful to make myself and also go over the terms. I thought it really helped me understand the terms and the process together as a whole. And yeah, thank you guys so much for watching. I hope you have a wonderful rest of your day. That is the end of my presentation. Have a good one.